I'd like to introduce our next Gilbreth lecturer, Dr. Caitlin Howell. Caitlin is Associate Professor of Biomedical Engineering and head of the Biointerface Biomimetics Lab at the University of Maine. Since 2016, she and her team have worked on drawing inspiration from nature and working across disciplines of biology, chemistry, material science, to address major issues in human health. Inspired by how the human body controls its large, diverse bacterial cohorts, her work has led to discoveries and methods to resist infection in medical devices without need for antibiotics. With an eye towards securing the health of our planet's citizens, Caitlin will discuss materials-based approaches to prevent biofilm-associated infections. Please welcome Caitlin Howell.
Hey everyone. It is so great to be here. I'm exceptionally honored to be able to talk with you today, and also a huge shout out to all the students here, because students, we're really gonna need your help with this one. Okay? All right. Who knows what this is? Yes. Oh, I love it. I love it. Okay, so if you know this is bacteria, you probably also know that bacteria are around us all the time. They are on our surfaces, they are in the air, they are on our bodies, they are in our bodies. Most of the time, we don't notice them, right? They don't bother us, they don't really do much, we're way too small to see. Sometimes they do really good things, right? Like when they make us cheese for pizza, and the ones inside of us that help us digest that wonderful cheese pizza. But when they decide to not do nice things, it can get really ugly, really fast. Bacteria make us sick. They can cause infections, and this is a really big problem. However, luckily, we have basically what amounts to a magic medicine, antibiotics. Raise your hand if you've ever used antibiotics. Everybody in this room, at some point, we've had an ear infection, we've had strep throat, we've had something. And if you've taken antibiotics, you know that they really are magic. You take them and you start feeling better, and it's awesome, right? Well, I got some bad news. Slowly, little by little, they're not working so well anymore, right? Antibiotic resistance is a real problem, and folks, it's coming. But before we get too much into that, let's go back to a little bit of basic biology and talk about why this is happening. All right, consider, if you will, a population of green bacteria. Now, these green bacteria, like all living organisms, are made up of genetic material, which is a little bit different from cell to cell, right? We got some mutations in there, this is a thing. But we don't like these bacteria. We want them to go away. And so we're gonna hit them with green bacteria antibiotics. Awesome, right? So this works really, really well, and most of the cells die. However, we had a few cells in there in that genetic mix. Because of the diversity, we're just a little bit purple. And it turns out that green bacteria antibiotics don't really work so well on purple bacterial cells. But most of the cells are gone, so we don't really notice. We think the problem is solved, and we go about our daily lives, and that's great. Until some time passes, and suddenly the bacteria are back. But this time, we got a lot more purple cells. Maybe we don't know this though, because we can't really see these things very well. So we're just gonna hit it again with some green bacteria antibiotics. And the problem gets worse and worse and worse, right? Yes, sound familiar? So you may think, well, yeah, sure, but when is this actually happening? The answer is it is happening all across the world every single day. That patient that gets antibiotics prescribed to them for an infection, starts taking them, feels better, and then decides, I'm better. I don't want to take medicine if I don't need it, right? That's the healthy thing to do. I'm going to stop taking my antibiotics. Antibiotic resistance. It's that farmer, right, who knows that if he or she feeds their chickens with antibiotics, those chickens will get fatter. Because it turns out if you kill the bacteria inside your chickens, the food that you then feed those chickens goes into making more chicken and less bacteria inside of chicken. Yes, it really is a thing, and that is happening more and more and more. They just came out with a report talking about how this problem is increasing. It's also those factories that are making these materials. And if they happen to be in a place where the environmental restrictions aren't so tight, Maybe they're just dumping that waste into the environment where it goes out and it affects the bacteria in the environment. Antibiotic resistance. Every single little one of these examples is a tiny drop in the bucket. But folks, we've been dropping into that bucket for the past 60 years, and now it's starting to get full. And the result is really, really devastating. Right now, we are living in the post-antibiotic world. Around the world, each year, 5 million people are dying of antibiotic-resistant infections. And of those people, heartbreakingly, most of them are children under 5 years old. It's really, really terrible, and this is just the beginning. It's going to get worse and worse and worse. So, are we doomed? No, not as long as there is an engineer standing, right? We solve problems, and that's what we're going to do. Now, it's not going to be just engineers that solve this problem, though. It's going to be a huge team effort. We're going to need scientists. We're going to need medical professionals. We are going to need communities. It's going to be all of us in this together figuring out this solution. And the solution is not going to be just one-sided, right? It's going to be a lot of different avenues of attacking this. But at my group, the University of Maine, as you heard, we study bio-inspired solutions. And in particular, we focus on materials because it turns out that bacteria really grow really, really well when they're sticking to surfaces. And so we're focusing on that. Now, bio-inspired engineering, for those of you that don't know, is really great. It's looking at nature for the way that nature solves problems and then stealing those ideas and using them for human problems, right? Okay, so please, if you would, consider this beautiful, idyllic scene. There are bacteria everywhere in this scene. They are in the cows, they are in the soil, they are on the plants, they are in the air, they are everywhere. But this is not a rampant bacterial apocalypse. You don't have infections happening everywhere in this scene. Neither do you have massive use of antibiotics. So what's happening here? The answer is there are a lot of things happening in this picture that is helping to control those bacterial populations. But I could spend a year telling you about all the amazing things that's happening, and interestingly enough, there's a lot of ways that we still don't know how nature is doing this, so there's a lot here to discover. But for the sake of time, I'm just going to walk you through 3. These ones. I'm going to talk to you quickly about dragonfly wings and cicada wings, also lotus leaves, and also that last one, which I think you had a hint about earlier, but I'm not going to tell you about it until we get a little bit later. All right, cicada and dragonfly wings. So we didn't know this until 2012, but actually it turns out that dragonfly and cicada wings are covered with nanospikes. Did y'all know that? No? Now you do. And a really, really interesting thing happens when bacteria interact with these nanospikes. They pop. If you take a bacterial cell and you push it down on top of these nanospikes, that bacteria cell will rupture. And what you get is basically a bunch of deflated balloons. This is really cool, huh? An amazing way that nature handles this. However, if we're thinking about not only identifying these solutions, but applying them to human problems, we got to think about whether or not this is actually going to apply to our problems with bacteria. So let me ask you a question. Once a bacteria goes pop on this surface, and then another bacterium comes along and sits on top of it, is the same thing going to happen? No. So this solution works great for dragonflies because there's not that many bacteria coming down to land on their wings, and also they're not really using their wings for that long. They don't live that long. So great solution for dragonflies and cicadas and maybe some short-term human use, but not really for the things that we really need. Okay, we need something that maybe is a little bit more replenishable, something that can be rejuvenated. So let's look at the lotus leaf. Did you know that the lotus leaf has a really, really interesting way of keeping its surface clean? Turns out that if you look really closely on the surface of this lotus leaf, it has a series of bumps. And those bumps have a series of even smaller bumps on them, nanobumps, if you will. And all of these lovely bumps are covered with a material that resists water. It's hydrophobic. Okay, so what happens if a water droplet comes down and sits on this surface? Instead of sort of sticking and flattening out a little bit like it would on a flat surface, this water drop will actually sit on top of those bumps. And in sitting on top of those bumps, it is also sitting on top of those pockets of air in between the bumps. You know, water and air don't mix very well. The water doesn't like to be sitting next to the air. So when any sort of nice breeze or bump comes along, that water droplet can actually start to roll. And it can pick up any dust, dirt, bacteria along with it and roll right off. So this is great, right? Hey, here's a cool solution, replenishable. Oh, there's a trick. This only really works if you're interested in resisting water under ambient temperature and pressure. So under usual lotus leaf conditions, right? Probably not what we're interested in. When we're interested in resisting bacteria on materials, the stuff the bacteria is in is full of proteins, it's full of lipids, it's full of all kinds of gunk that is not gonna do what this beautiful water droplet does. So maybe we're looking in the wrong place. Maybe we need to look a little bit closer to home. I see lots of inspiration in this room right now. Did you all know that you have more bacterial cells inside of you right now than you have you cells? You are not just a human, you are a superorganism of bacteria and human. And it's pretty cool because we're learning every day just how much our bacteria affect everything we do, up to and including how we think. But think, if you will, about the place in your body that has the most bacteria. Anybody know what this picture is? I think I'm hearing it. I'm hearing lots of great answers, and you're all really close. Intestines, yeah? Okay, we're close. Intestines. You have so many bacteria in your intestines, and this is a beautiful picture. I did not take this picture. This was taken by Lars Verrike of Ghent University, but I'm such a fan of it because it shows so beautifully really how the body is doing this. So here, up here, this Las Vegas party, those are all the bacteria. Down here, you have your beautiful, fragile, susceptible intestinal cells. And the thing that is stopping these bacteria from getting to these cells and having a field day is your mucus. Now, if you walk away with nothing from this talk, I want you to walk away with the fact that your mucus is incredible. We think it's gross when we first start out, but now that you've been here, you can know that it is an absolutely amazing material. It is liquidy. It moves, it slides, it sloughs off, it replenishes. And now that we've actually started studying a little bit more, we're starting to see that it actually chemically communicates with the bacteria that it's interacting with. It can literally take a bacterium that wants to turn into something that could infect you, and it can send it chemical signals that says, no, hey, chill, man, you're cool, you're good. Go into that nice dormant state, you're happy, you're fine. It's really, truly amazing. So as someone who does bio-inspired engineering, material science, I look at that and I go, okay, we want to copy that. That is what we need. How do we do it? There is no way we're going to be able to mimic the beauty of mucus in its entirety. But what we can do is get close. So let's think about, at the very basic fundamental level, what is that doing? It's stopping the bacteria from getting to the cells. Let's see if we can copy that. Turns out we can, fairly easily. So in my lab, we're actually doing that on a material that is very widely used in medicine, silicone rubber. Clear, squishy stuff. It's everywhere, in tubings, on machines. It gets implanted in people, and it's a great place for bacteria to grow. So it turns out, though, that we can make a mucus-mimicking layer on this simply by immersing this material in a bath of a compatible liquid. And when it does, this silicone rubber acts like a sponge. It sucks up all of that liquid, and when it's done, we end up with an infused polymer, an infused silicone rubber. It is that material infused with our liquid with another layer of liquid on top. You seeing the parallels with the intestines and the mucus here? All right, so let's try this. So we took this stuff into the lab, and we tried it against one of the nastiest bacteria there are. This is called Pseudomonas aeruginosa, causes huge problems in hospitals. So we actually took a bath of this stuff, and we treated a piece of tubing. We stuck this tubing in, and we allowed the bacterial solution to pump around that tube over and over and over for 24 hours. Then we took it out, we stained the bacteria purple, and we saw what we had. Now in the picture I'm going to show you next, we actually treated half of the tube, and we left the other half of the tube untreated. Can you tell which is which? Yes, you can. Here, where we had no liquid layer, we had lots of bacteria growing. These things make beautiful, beautiful, thick, slimy biofilms, they're called, bunches of bacteria. On the part that was untreated, when we looked at it, well, we saw a couple of glowy green things there, but really, really not much. So this was very exciting. We thought maybe we have something here. But in biomedical science, anyone can do anything on a lab bench. But all bets are off when it comes to a living system. You are so beautifully complicated that we could never in a million years mimic all of the stuff that happens inside of you all the time. So the only way to know if something really works is to try it in a living system. So at the University of Maine, I don't have the capacity to do that. But what I do have the capacity to do, and you all will as well, is make science friends. And I made a wonderful science friend at the University of Notre Dame. Her name is Professor Ana Flores-Morales, and she's one of the leaders in studying what are called catheter-associated urinary tract infections. Raise your hand if you know what a catheter is. Okay, for those of you that don't, it's one of the most common medical devices. It is a little tube that is inserted up through the urethra into the bladder, and it allows urine to drain out for folks who cannot control that function, either because they've lost the ability or because they're under anesthesia. Very, very common. A huge source of infections. And my science friend, Dr. Flores-Morales, studies this. And she had actually found that what causes the bacteria to stick so well to these devices was not actually the bacteria themselves, which is one of the most common assumptions right now. She told me, Caitlin, you know what it is? It's the proteins. You have proteins floating around inside of you all the time. And when you put a foreign object inside of you, the first thing that happens is proteins stick to it. And then the bacteria stick to the proteins. And that's what is causing the massive problem. So once these bacteria start to stick to these medical devices inside the bladder, the bacteria start to grow, and then they do something called dissemination. They spread to other organs. And that's when it gets really, really, really bad. Now imagine this situation with no antibiotics to solve it. Yeah. So she asked me, can this system also resist proteins? What do you think? Yes. There's no reason why it shouldn't, right? It's a slippery mucus mimicking surface. Proteins are not also sticking to that mucus layer. So we designed an experiment. We were actually going to try our treated catheters, our liquid-infused catheters against this, and our hypothesis was that we would resist bacterial adhesion, and in doing so, we would resist dissemination. So here's a quick view of the results. This looks complicated, but let me walk you through it step by step. First, look over on the right there, just those pictures. There you can see fluorescent images. So we have protein sticking on the top. You can see our unmodified catheters, and then you can see our liquid-infused catheters. Can you see the difference? Where it's glowing, there's protein. The CTL there is our control. You've all read about controls and experiments, right? Yes, very important to make sure the system is working. E. coli, you've all heard of that? Massive problem in urinary tract infections. Again, where it's glowing, we've got bacteria. Where it's not, we don't. And then we actually put these two images on top so you can see if there's one where there's the other, and in fact there is. So already we were seeing this is what's having an effect, again, in a living system. And this plot on the left here is where you see that quantified. So here we have the amount of bacteria, CFUs, colony-forming units. How many bacteria are there? I want to call your attention to the fact that this is a log scale. This is an order of magnitude every way up. Here we have the different organs, and you have unmodified and liquid-infused. You see the drop there when we have that liquid-infused? Now that doesn't look like much, but again, order of magnitude, that's a 90% drop. Yeah. These little bars up here means it was very statistically significant. And you saw we got that across the board. Non-significance right there just because the bacteria hadn't gotten that far. These were for us jaw-dropping results for a living system. It was extraordinarily exciting, but even more exciting, we tried this against all of the top 6 pathogens that cause these types of infections in hospitals, and they worked against all of them, including this one, Candida albicans. It's a fungus, folks. They're even worse because we have even fewer medicines that help against them. So this was really, really exciting. And I think it shows that if we start thinking about the power of nature, we can really start getting places. But I want to take you one step further. Okay, consider this material. It's static. Wood, oh, awesome, on top of it. It's static. It sits there. It's wood, but it's not a tree, right? It's dead. It does what we need it to do in this situation, but it doesn't heal. It doesn't respond. It doesn't sense. It doesn't do anything like that. Maybe when we're thinking about materials, we're thinking a little bit too much like humans still. Consider your materials. You can sense things. You can heal. You can respond to your environment. If we're talking about putting things into a living system, why would we want this and not this? Yeah, let's start thinking out a little outside the box, huh? All right, so let's take our favorite example here. And what is making this possible? Again, a lot of things. You're really complicated. But at the very basis, it's your vascular system. Your vascular system is carrying information and nutrients to your cells. It is taking away information and nutrients from your cells. It allows the whole system to keep going and going and going. So why can't we have vascular systems in our materials? We can. Thanks to 3D printing, which we're going to hear a lot more about, we can build vascular networks into materials, and so we did. We did this experiment where we built our vascular networks, and then we filled our vascular networks with our slippery mucus-mimicking material. And the concept was that when that layer was removed, more would come up from the vascular system and replenish. And we did this experiment where we actually removed our liquid layer over time, and you can see this is the amount of bacteria. Our dry surface, it starts out low and then it goes up and up and up. By the time we've removed most of that liquid, we're back to the same old surface. Now when we put a vascular network in this, same samples, side by side, suddenly these can resist the bacterial attachment for the entire process of the experiment. But we can go even further. As I said, our vascular networks communicate with our tissues and can respond. So we said, let's do the same thing. We want to use our vascular network to listen to the bacteria. Let's hear what they're telling us, right? We need to know what's there in order to address it effectively, and then let's use it to respond. Let's control those bacteria, and let's do this in a loop constantly so we can act like a natural system. So we set up an experiment where we just listened to the bacteria. Here is our vascular network, and our hypothesis was that when the bacteria grew on the surface, the compounds that they constantly produce would diffuse down into our vascular channels. We could collect them and see what was going on. This is how bacteria grow over time. You can see we get more and more, and then they start to get static. We could do that by measuring on top, and when we looked in our vascular system, we saw the same thing. We used 2 different methods of being able to test this. This was a very cheap absorbance measurement, matched that curve. And here, this is a more expensive measurement, but we found something we weren't expecting. Not only did we see the bacteria growing, we could also see them eating their food. So here we go. We could listen to the bacteria. Now, can we control them? Well, to do this, we can do a neat trick of bio-inspired engineering, which is we're not defined by the same constraints as nature. We can actually define things and change things to better suit our needs. So what we did was actually change the way our vascular network looked like. And I asked my students, hey, can we make the bacteria grow in dots? And it turns out we could. By controlling where those channels were and putting into that vascular network a material that told the bacteria to stop growing, where that material rose to the surface, we got bacteria dying. And where that material didn't reach, we got bacteria living. And so here was our theoretical experiment, and here was our theoretical calculation, and here was our experiment. So the take-home message, besides the fact that mucus is amazing, is that we can learn a lot by looking at nature. The antibiotic resistance crisis, we can solve it. There are a lot of ways that we can approach this, but this one, looking at how natural systems that have evolved over millions of years are solving this problem already, we can learn a lot from them, and we can go forward into a future where we don't have to worry about this quite so much. All right, that, my students, of course, and my funders, and thank you all for your attention.
Okay, we have questions.
You can ask whatever. What? Oh, it was already answered. Okay. Oh, awesome. That's great. Yeah.
All right, Vicente. Okay. All right, Vicente Valenzuela Jr. from Cabrillo High School. So going back to the beginning of the presentation with insects, how did you guys know that dragonflies were going to be the insect that works when there's butterflies, beetles, and a regular fly all with wings?
That is an excellent question. So that was not my work. That was coming out of Canada and a collaboration with Australia. But the answer is we didn't know. This is the cool thing about bio-inspired engineering. There's still so much to explore in nature. And I think someone was just looking at how these things were structured and probably happened to see a bacterial cell on there and went, wait a minute, what the heck is happening here? And then started to investigate further. This actually happens a lot. And so we get a lot of the really best inspiration as engineers from those crazy biologists that go out and they're just like, I wonder what this looks like. I wonder how that works. And then they're collecting this information. It's really a synergistic process. But that's a fabulous question.
I'm Xavier from Cabrillo. Yeah, but I was gonna say, how did y'all stop making the bacteria reproduce?
How did we make the bacteria reproduce?
How did y'all stop making it reproduce?
How did we stop making the bacteria reproduce? That's a great question. So we weren't actually stopping them from reproducing because in a living system we wouldn't be able to do that. So the thing that we were doing was actually just stopping them from adhering. And it turns out that when bacteria adhere to a surface, that to them is like being in a really, really nice place. They start to build a little house around them, if you will. They start to excrete these materials, these polymers, and they cover themselves with it. Have you all seen in a sink where it gets slippery when you have bacteria start to grow and it's gross? That's because what's growing on the surface is not just bacteria. It's bacteria plus all that stuff they pile around them to make them feel nice and protected. And the bad thing is that that material, it's called a biofilm altogether, that can actually stop antibiotics from penetrating down to get to the bacteria and kill them. And it's another reason why we study materials, because they don't do that unless they've got a material to stick to. So that was an excellent question. We didn't stop them, but when we stopped them from sticking, we stopped them from building those biofilms, we naturally stopped them from growing as much as they would have anyway.
Hi, my name is Viviana Valenzuela. I'm a sophomore at Cabrillo High School, and I was wondering, do you think that you could put your work into everyday hospitals?
Yes, actually, that's an excellent question, and we are currently in the process of moving this through FDA approval. The Food and Drug Administration, the entity responsible for making sure that everything that gets to you in terms of medicine is safe and effective. We're starting the process of working with them and working with several companies, because this is really a game changer, and a lot of people recognize this. So, excellent question.
Hi, my name is Mia Barca. I'm a junior at Samuel Academy. This is kind of related to her question, but I was wondering if you think at some point in the future you'll be able to create a synthetic mucus that will make hospital infections obsolete?
I love that question. I really hope so. That's my goal as someone who does bio-inspired engineering, right? I aspire to be able to mimic the beauty that is mucus, but it's really step by step. And it takes, as we've heard before, it takes a lot of different people with a lot of different disciplines working together to make this possible, but I really hope that I'll be able to be a part of that puzzle, because it would be extremely exciting, because there's a whole lot more we could do, right? If we could control bacteria and make them do what we wanted them to do, we could go crazy. Great question.
Hi, I'm Andrew Percussion, senior at Cabrillo. With the introduction of this technology to hospitals, could this affect like sterile processing technicians and how their jobs will be?
Oh, that's a really good question. So I don't think so, because this is something that it is a coating that is meant to be put in one particular place. I think at no point in the future are we looking at being able to replace like surgical instruments with this type of stuff. And so sterilization in that sense is still going to be the same. We still need a way to sterilize gowns, to sterilize masks. All of these variety of materials that are used in hospitals will still stay the same. But it's really just those materials that are really causing a problem and infection that we're looking for solutions for because of the drive towards antibiotic resistance. Does that answer your question? Okay, thank you.
Hi, my name is Sam. I'm a senior at Cabrillo. My question is, so with whether it's the mucus or the what was it, like you kind of copied the mucus, or whether it's anything else, do you see it moving to like maybe everyday cleaning supplies, or like how she said, like the hospitals and stuff? Like, what do you think?
That is a really, really good question. And I think this comes to a really interesting concept in materials engineering, is that you really have to consider where it's going to be used. So if I were to be able to design the perfect synthetic mucus, I would probably never put it on something like this, because it's slippery. You can remove it if you slide it around enough. It wouldn't be perfect for all situations. In our bodies, mucus is really only used in the one place where there's just a lot of bacteria and a great need, and where it's sloughing off a little bit is not going to be a problem, and nobody's touching it and rubbing it. So I think every time we think about designing a new material or a coating, we need to consider that. Where will it be used? How will it be used? And does what it looks like make sense in that context? Does that answer your question? Okay, thank you. That's a great one.
Hi, my name's Michael Bradfield from Cabrillo High School. I'm a junior. I was wondering if you used a bacterial prevention in dental care, maybe? Like, could we use it on our teeth, or like to prevent plaque buildup, like a sticky web, or even the coating that you were using?
That's also a great question. There's actually somebody who did a study on that. It wasn't me, but they tried to put it on rabbit teeth to see if it would stop the buildup of bacteria. And in fact, it does. But there's a trick. You actually have to be able to treat the surface of the teeth to make that coating stick. So in the materials that I use, the coating sticks naturally because it's very compatible with the material that we're using as the base, the substrate. But in teeth, you would need to make the surface rough, and then you would need to apply a sort of chemical coating over that to make it stay put. So while it's possible, it might not be particularly practical, at least not in its current form. But I think there's a lot of capability now that we know that this approach can work to think about, well, how can we change the design to be able to make it work in a place like that? And that's another thing we could all use your help with. So, got any ideas?
Let's do one more question.
We have one question. Hi, my name's Eric. I'm from Somali Academy, and I'm a junior here. I would like to ask about, the fish that have mucus on them. Have you ever tested that?
Yes, there actually have been lots of studies looking at that type of mucus and what it does and how it helps, and ideas about how we can use that to make, for example, ships go faster. Because what that really does is it reduces the friction against the water, which we may think that, water, you just slide through it, it's no problem, but it actually does produce drag. It slows things down. So people have looked at that, yes, and that's another excellent example of let's think about nature and what does the thing that we'd like to sort of kind of do. And let's see if we can take that idea. So already you're on the path. Great question.
One final question, maybe? Anybody? Somebody over here?
I had a question. Is there any way that we could use mushrooms in biomedical?
In mushrooms in biomedicine? Yes. So you mean like the materials of mushrooms, or you mean using mushrooms as medicine?
Oh, okay. No,
We don't want to specify. Yes, the answer is absolutely yes. And I think we're actually going to be looking a lot more at mushrooms lately in the context of this. So do you remember at one of those I pointed when I was talking about the bacteria that the system worked against, and I said this one's a fungus, that's really bad. Do y'all know why fungi are so much more difficult to control than bacteria? Spores, yes, but fungi are actually pretty closely related to you compared to bacteria. So anything that will kill a fungal cell will also kill a U cell. That's part of the reason why it's so difficult to get rid of mushrooms, right, of fungi. So, however, in the kingdom of fungi, there are a lot of control strategies that they use. So I think we're going to go in the direction of looking at those to see if we can find solutions to our current fungal problem. Thank you for your question. All right, are we good?
Thank you very much. I have, Caitlin, I have two things to say to you. One is if you can bottle that enthusiasm, you'll get rich. Okay, we can all use it. And secondly, I'm not going to look at my amazing mucus in the same way as I go forward. So thank you.